Hello and welcome to some biology class. Friends, today we shall discuss about fermentation. Fermentation is the enzyme catalyzed anaerobic breakdown of energy-rich compounds such as carbohydrates. It is a biological process. Pyruvate is the end product of glycolysis. Under aerobic condition, pyruvate is completely oxidized to carbon dioxide and water via citric acid cycle. Whereas under anaerobic condition, pyruvate must be converted to a reduced end product in order to reoxidize the NADH produced by glycerol di3-phosphate dehydrogenase catalyzed reaction. This occurs in two ways. Under anaerobic condition in muscle, pyruvate is reduced to lactate to regenerate NAD+ in a process called as homolactic fermentation. In yeast and certain other organisms, pyruvate is first decarboxylated to yield carbon dioxide and acetaldehyde, which is then reduced by NADH to yield NAD+ and ethanol in a process called as alcoholic fermentation. In aerobic glycolysis, NADH acts as a high energy compound, but in anaerobic glycolysis, its free energy of oxidation is dissipated as heat. Homolactic fermentation. In muscle during vigorous activity, when the demand for ATP is high and the oxygen is in short supply, ATP is synthesized largely via anaerobic glycolysis, which rapidly generates ATP rather than through the slower process of oxidative phosphorylation. In this process, commonly known as homolactic fermentation, pyruvate is reduced by NADH to yield NAD+ and lactate. This reaction is catalyzed by the enzyme lactate dehydrogenase. Lactate represents a sort of dead end for anaerobic glucose metabolism. It can be exported from the cell or it can be converted back to pyruvate, as this reaction is reversible. In skeletal muscle, much of the lactate produced during anaerobic glycolysis is carried via blood to the liver, where it is used to synthesize glucose. This equation represents the overall process of homolactic fermentation, in which one glucose molecule is partially oxidized to yield two molecules of lactate, and there is a net gain of 2 ATP. Alcoholic fermentation. In yeast and certain other organisms, under anaerobic condition, pyruvate is first decarboxylated to yield carbon dioxide and acetaldehyde. This reaction is catalyzed by the enzyme pyruvate decarboxylase, for which thiamine pyrophosphate is the essential cofactor. In the second reaction step, acetaldehyde is reduced by NADH in the presence of enzyme alcohol dehydrogenase to yield NAD+ and ethanol. This process is commonly known as alcoholic fermentation. This equation represents the overall process of alcoholic fermentation, in which a molecule of glucose is partially oxidized to yield two molecules of carbon dioxide and two molecules of ethanol. As in homolactic fermentation, in alcoholic fermentation also there is a net gain of 2 ATP. So friends, this is all about fermentation. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much.